ब्रह्मनाथ ओ शांति 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 स्वस्थ।
Hello students, in the previous class we discussed about the immunity. What is the immunity system? Immunity system, it is a self-defense mechanism in which it prevents against the foreign particle, or we can say the pathogens. Further, we discussed immunity is of two types. One is your innate immunity, other one is your acquired immunity. Innate immunity, it is also known as your non-specific immunity, whereas the acquired immunity, it is also known as your specific immunity. Innate immunity is present since birth, whereas the acquired immunity, as it is acquired, means it is present after the birth. Innate one is your non-specific because it is not specific to a particular pathogen. It can kill or it can fight against any pathogen. But the acquired, the immunity, it is the we can say it is the specific one. Okay, means it is specific for some pathogen. But the innate immunity, it is non-specific, means it is not specific to any pathogen. It can kill any pathogen. Further, we done the division in which we discussed the innate immunity is divided into two types. One is your external immunity, other one is your internal innate immunity. External innate immunity, we discussed clearly the further division in which we discussed the physical barrier and also the physiological or the chemical barrier. Today we will discuss the internal innate immunity. Okay, internal innate immunity, which act as the second line of defense. Okay, what is the first line of defense, second line of defense, third line of defense? As the external innate immunity, it is the first line of defense, means whenever the pathogen enter into our body, first it will fight against them, means first that the line of defense will fight against the pathogen. If it is unable to fight, then the second line of defense will come into act. If second line of defense will fail, then the third line of defense, which is your acquired immunity, which will come into act. Okay, so your external innate immunity is your first line of defense, whereas today we will discuss the internal innate immunity, which is your second line of defense. Okay, under the internal innate immunity, first we will discuss the cellular barrier, or we can say we will discuss firstly the phagocytosis. What is phagocytosis? Phagocytosis means it is a process. It is a process in which the phagocytic cell engulf the pathogens, various cellular debris, etc. Whenever any foreign particles or any pathogen enter into our body from the outer environment, the phagocytic cell engulfs them, or we can simply say kill that pathogen. Okay, so we will write your first point. It is a process in which the phagocytic cell engulf the pathogen cellular debris, etc. Okay, next point, phagocytosis. We can say it acts as the second line of defense, as I said earlier. It acts as second line of defense. Okay. Third point, what are various phagocytic cells, such as your neutrophils, monocyte, which are the various types of WBC, then the tissue macrophages. Okay, various phagocytic cells are such as your neutrophil, monocytes, okay, which are your types of WBC, we can say. Okay, other one also there, such as your tissue macrophages, etc. Okay, etc. They are common phagocytic cells. Acha. Now come to a concept how the phagocytic cell comes to act. Suppose we'll see, suppose this one is your phagocytic cell. Okay, suppose this one is your phagocytic cell. We'll not go into the detail of the content or what are the means the internal portion that are present in the the organelles that are present in the, the phagocytic cell. We'll just focus on the lysosomes, okay, that are present inside the phagocytic cell, which helps in digesting the pathogens. Suppose this is your phagocytic cell. Suppose this one is your phagocytic cell. Okay. Suppose this one is your bacteria. Suppose this one is your bacteria. Okay. This one is your bacteria. Whenever bacteria, or we can say pathogen, or bacteria, or we can say pathogen or bacteria. Okay. Pathogen or pathogen, you can simply say pathogen, or we can say bacteria is, in this case we consider the bacteria. Suppose the bacteria enters into our body, by which the phagocytic cell gets activated, okay, by which firstly it will engulf the bacteria, okay, by which the intracellular vesicle or phagosome is formed. Firstly, the intracellular vesicle or phagosome is formed. Okay. When the lysosome fuse with the phagosome, then the phagolysosome is being formed. Phagolysosome is formed. In the phagolysosome, the lysosomal enzyme helps in digesting the pathogen, or simply we can say kill the bacteria, forming the digestive vacuole. Okay, forming the digestive vacuole. Digestive vacuole. And after the pathogens are killed, the residual bodies are being eliminated. The residual bodies are eliminated. Okay, so this one is your simple concept in the phagocytic cell. Okay, and some common examples are your Kupffer cells in the liver or the glial cells in the brain. Common examples are Kupffer cells in the liver. Okay. Glial cells in brain. Okay. Okay. So further till now we discussed what is your phagocytosis. Means it is a process in which the phagocytic cells, okay, engulf the, we can say, the pathogens or the cellular debris, etc. So now then we discuss what are your various common phagocytic cells such as your neutrophils, monocytes, which are your types of WBC. Then we discuss such as your tissue macrophages are also a phagocytic cell. Then we discuss what is the concept means. First, suppose this is your phagocytic cell, and whenever the pathogen, suppose here we have taken the bacteria, enters into our body, the phagocytic cell gets activated and it engulfs, and as soon as it gets activated, it goes near to the bacteria, by which firstly it engulfs the bacteria or the pathogen, forming the intercellular vesicle or the phagosome. Lysosome or the lysosomal enzymes are present. The lysosome fuse with the intercellular vesicle or the phagosome to form the phagolysosomes. Okay. In the phagolysosome, the lysosomal enzymes kill the pathogens, or we can say digest the pathogen, forming the digestive vacuole. And the remaining portion or the residual bodies are being eliminated. And the common examples are copper cells in the liver, glial cells in the brain, etc. Okay. So this is all about your phagocytosis. Please note down. Now come to your inflammatory barrier. Okay. First point, in the case of the inflammatory barrier, it is a non-specific response which develops due to the injury of the tissue or its infection. Okay. Whenever tissue injury happens, then in that case the response is created which is known as your inflammatory barrier. So we can say it is a non-specific response. It is a non-specific response which occurs due to the injury of the tissue or its infection Okay. Second point, it is a localized response, means in which place the tissue injury happens, only the response occurs only confined to that area only. It does not spread to any other area. That is why it is also called the localized response. It is a localized response. Okay. What are various signs we see? Redness, swelling, increase in temperature, pain, etc. Redness, the signs we see are such as your redness, swelling, increase in temperature, pain, etc. Okay. Now come to your concept, how the inflammatory barrier comes to an act. Suppose this one is your tissue. Okay. Suppose this one is your tissue layer. This one is your blood vessels. Okay. Suppose this one is your blood vessels. Okay. This one is your tissue layer. This one is your blood vessel. And suppose this one is your portion where the tissue injury happens. Okay. Tissue injury happens. Whenever the tissue injury happens, the cells, means the mast cell which are present inside the tissue, breaks. These are your mast cell. Okay. The mast cell breaks. Okay. And secrete a vasodilator secretion known as your histamine. Secretes vasodilator, secrete histamine, which is a vasodilator. So vasodilator means it simply helps in dilating the blood vessels. See, whenever tissue injury happens, the mast cells break, they secrete the histamine which acts as a vasodilator, which helps in dilating the blood vessel, which helps in increasing the blood flow, by which the redness occurs. The plasma and the fibrinogen from the blood vessels leak out into the tissue, and the tissue fluid also accumulate, by which swelling occurs, okay, and the swelling leads to the increase in temperature, okay, which will further lead to the activation of the phagocytic cell and simply kill the pathogen. Increase in temperature will activate the phagocytic cell, and further the phagocytic cell will engulf the pathogen. So the inflammatory barrier we discussed, it is a non-specific response occurred due to the tissue injury or its infection. It is a localized response, means it is confined to only one area. Various signs are seen. Firstly, we can say the redness, the swelling, the increase in temperature, and obviously the pain will be seen. Concept means this is a tissue. Further, if in this portion the tissue injury takes place, the mast cells which are present in the tissue will break down. By further, which secretes histamine, which acts as a vasodilator. As it is a vasodilator, it will helps in dilating the blood vessel, by which the increase of the blood flow takes place, by which the redness is being seen. Okay, and the fibrin and the plasma and the fibrinogen which leaks out, which leaks into the tissue, and the tissue fluid accumulates to form the swelling. Okay, and the swelling will lead to the increase in temperature, and that increase in temperature will help in activating the phagocytic cell, which will help in engulfing the pathogen, or we can simply say it will help in killing the pathogen. Okay, please note down. Now come to your writing part of the inflammatory barrier. After the tissue injury, the mast cell breaks. First point. After the tissue injury, the mast cell breaks. The mast cell secretes histamine and the prostaglandins. So simply you write the mast cell secretes, generally we will write the histamines, okay, which act as a vasodilator, which acts as a vasodilator. Okay. What does the histamine? As it acts as the vasodilator, it increase or dilate the blood vessel, by which the blood current, means the flow of blood, increases. The histamine dilates the blood vessels. Histamine dilate the blood vessels. As a result, the blood flow increases. By which the redness occurs. Okay. Next point. The plasma and the fibrinogen leaks out into the tissue, and the tissue fluid is also get accumulated, by which swelling occurs. Plasma and fibrinogen leaks out to the tissue, and the tissue fluid accumulated, resulting the swelling. Okay. Further, the swelling leads to the redness and the swelling leads to the increase or we can say rise in the temperature. And the rise in temperature helps in getting activated the phagocytic cells. Further, the phagocytic cells engulf or kill the pathogen. Rise in temperature activates the phagocytic cells, okay, which further engulf the pathogen, Okay. So in this manner, this is your writing portion of the inflammatory barrier, or we can say the inflammatory barrier comes into act. First, the tissue injury happens, and when the tissue injury happens, the mast cells break down and the mast cell secretes histamine and the prostaglandins. Here we are writing the secretes the histamine, which act as the vasodilator. As it is the vasodilator, it dilates the blood vessel, and also as a result which the blood flow increases and the redness occurs. The plasma and the fibrinogen from the blood vessel comes out or leaks out to the tissue, and the tissue fluid are get accumulated, forming the swelling. Swelling and the redness leads to the increase or rise in the temperature, and the rise or the increase in temperature helps in getting activated the various phagocytic cell. And as soon as the phagocytic cell gets activated, they engulf the pathogen. Okay, please note down. Now come to your elevated body temperature or fever. So what is elevated body temperature or fever? Sometimes the rise in the body temperature is good, which is useful for our body, it acts as an immunity booster, it activates our immunity system. But more increase of the body temperature, it is bad. Okay, so we can say what is your elevated body temperature or fever? Whenever pathogen enters into our body, okay, it secretes various toxic material or toxins. Whenever the toxins are being released, then the various cells such as your monocytes and macrophages secretes various secretion called the endogenous pyrogen and also the interleukin. Okay, then when the secretion are being done, the endogenous pyrogen and the interleukin, the pyrogen reach to the brain, okay, specifically to the hypothalamus region. Whenever the enough pyrogen reach to the brain, or we can specifically say the hypothalamus region of the brain, then the body temperature increases. As a result, elevated body temperature or fever occurs. But as we know, as the temperature of the body increases, the phagocytic cells get activated, okay, by which as the rising temperature also occurs, the growth of the various pathogens is also slowed down, or we can say the growth is totally stopped, or we can say unable to grow. And further, that the pathogens are being engulfed by the phagocytic cell. Okay, so now first point we'll write when pathogen enter into the body, it secretes, the pathogen secretes, okay, body pathogen secretes toxins, okay, pathogen secretes toxins. As soon as the toxin are being secreted, it gets activated various cells. Release of toxins activates various cells such as monocytes, macrophages, etc. Okay, which secrete endogenous pyrogen and interleukin. Okay. The monocyte secretes the pyrogen and the macrophages secrete the interleukin. Next step, when the enough pyrogen reach to the brain region, specifically the hypothalamus region, the body temperature increases. When enough pyrogen reaches the brain, specifically the hypothalamus region, the body temperature increases. The body temperature increases. As soon as the body temperature increases, the pathogens are unable to grow, and further the phagocytic cells engulf the pathogen or kill the pathogen. Last point, the increase in body temperature the increase in the body temperature makes unable the pathogen to grow and engulf or kill the pathogens. Okay, so simply this is what about your elevated body temperature or what is your fever about. Means firstly, when the pathogen enter into our body, it secretes various toxins. As the toxins are secreted, it activates various cells such as your monocytes or the macrophages, which secretes the endogenous pyrogen and the interleukin. When the enough pyrogen reach to the brain region, or we can specifically say the hypothalamus region of the brain, the body temperature increases. As the body temperature gets increases, the pathogens are unable to grow further, and the pathogens have been engulfed by the activated phagocytic cells. Okay, so this is all about your elevated temperature and fever. Please note down. Now come to your natural killer cells, or we can say the NK cells. Natural killer cells simply kills the virus infected cells. Okay, let's come to a concept. Suppose this one is your natural killer cell. Okay, and this one is your virus. This one is your virus. This one is your virus. As soon as the virus enters into our body, the NK cells, or we can say the natural killer cells, get activated. The NK cells get activated, by which it secretes a secretion called perforin. Okay, which will helps in making the pore on the plasma membrane of the virus. Okay, by which the tissue fluid will enter into the virus. Okay, into the virus, by which the tissue fluid gets accumulated inside the virus, and finally the virus gets burst. Okay, and the cellular debris gets engulfed by the phagocytic cells. Okay, so what we can write this one for some points. Suppose the NK cells, okay, secretes perforin, okay, which makes pore makes pore. First one you can write the natural killer cells. Natural killer cells kills the virus infected cells. Okay, so see which here as the NK cells get activated, it secretes perforin which make pore on the plasma membrane of virus. Okay, for which makes pore on the plasma membrane of the virus, or we can say the target cell, or also we can say the tumor cell. Okay, by which as the pore happens, the tissue fluids gets enter into the virus. The tissue fluid enters into the virus, or we can say the target cell. Okay, further the tissue fluids get accumulated inside the target cell. The tissue fluid gets accumulated. As a result, the target cell burst or virus burst. The target cell burst, and the cellular debris gets engulfed by the phagocytic cells. Okay, so this is all about your NK cells or the natural killer cells. Suppose the suppose the pathogen enter into our body, and we can say it infect the cell which is known as a virus infected cell or the target cell. The NK cells secretes a secretion called the perforin, which makes the pore in the target cell. Okay, or virus we can say, or the target cell, by which the tissue fluids get enter into the target cell, and the tissue fluids gets accumulated inside the target cell, and finally it ruptured or burst. Okay, by which the remaining cellular debris gets engulfed by the phagocytic cells. Okay, now come to your acute phase protein. Acute phase protein. What are the acute phase protein? These are the proteins which simply inhibits the viral infection. So we can write these are the heterogeneous plasma protein, okay, found in blood plasma. Okay, how it works? Whenever the tissue invasion occurs, means anything that enters into the tissue invasion occurs, the macrophages and the neutrophils gets activated. Macrophages and neutrophils gets activated and secretes cytokines. What are the cytokines? Cytokines, we can say it is a group of protein found in the cells of the immunity which fights the pathogen. Secretes cytokines which further stimulate the liver to produce acute phase protein. Okay, the acute phase protein further helps in inhibiting the viral infection. Inhibits the viral infection. Inhibits the viral infection. Okay, so such like this the acute phase protein acts. Okay, so what are the acute phase protein? These are the heterogeneous plasma protein found in the blood plasma. First point, whenever the tissue invasion, means anything invade the tissue occurs, then the macrophages and the neutrophils gets activated, which secretes the cytokines. Means the cytokines are the group of protein, okay, group of protein found in the cells of the immunity, which helps in fighting the pathogen. Further, the cytokines stimulates the liver to produce the acute phase protein, which simply helps in inhibiting the viral infection. Okay, so this is all about your natural killer or NK cells and also the acute phase protein. Okay, this one is your target cell. Remember, okay, the virus that is infecting the cells. So this one is your target cell. Okay, means virus infected cells. This one. Okay, always remember the NK cells, they release the perforin. It makes spore in the virus infected cell. Okay, or we can say the target cell. We can simply write membranes on the virus infected cell. This one is simply we can write virus infected cell. Virus infected cell. Okay, or the target cell or the tumor cell, by which the tissue fluid enters in the target cell and the tissue fluid gets accumulated. Lastly, the target cell bursts and the cellular debris gets engulfed by the phagocytic cells. So this is all about an NK cells or the natural killer cells, and this one is all about acute phase protein. Please note that. Now come to your interferon. Lastly, the interferon. What are the interferon? Interferons are the antiviral glycoprotein which are secreted by the host cell when it is attacked by the virus. Okay, means when the host is attacked by the virus, it secretes a secretion of an antiviral glycoprotein which is known as your interferon. Now come to your concept. Suppose this one is your virus infected cell. Means it is a cell, or we can say the host cell, which is infected by the virus. When it gets infected by the virus, this virus infected cell secretes interferon, which is an antiviral glycoprotein. This interferon simply diffuse into the adjacent healthy cells. It simply diffuse into the adjacent healthy cells, by which it will activate the antiviral activity of the healthy cell, by which it simply prevents the multiplication of the virus. Okay, so remember always the interferons always diffuse into the adjacent healthy cells, by which it will activate the antiviral activity which will result to the prevention of the multiplication of the virus. Now, what are the various types of interferon? There are basically three types of interferon. One is interferon alpha, interferon beta, interferon gamma. Interferon alpha, it is also written as your IFN alpha. IFN alpha. Okay, in your material it is written INF. Please correct it by writing IFN alpha. I am repeating once again, in your material it is given INF. Please correct it IFN. Okay. So the interferon alpha, or it is also known as your IFN alpha, it is produced by the WBC. Whereas the interferon beta, or as the IFN beta, it is produced by the fibroblast. Whereas lastly, the interferon gamma or the IFN gamma, it is produced by the T lymphocyte. If we will come to your activity, then what happens? We know already, we discussed. Let's read in detail the activity of the interferon. The interferon simply diffuse to the neighboring cells. As I said earlier, it will not infect or diffuse into the virus infected cell. It will simply infect or diffuse into the adjacent healthy cells or the adjacent neighbor cells which are healthy. Okay, so the interferon simply diffuse to the neighboring cells, which will induce, if we will go into the detail, what happens, how it activates the antiviral property. So it induce to activate the ribonuclease 1 enzyme. What the ribonuclease 1 enzyme does? It breaks the mRNA of both the host and the virus cells. okay, which will eventually result in stopping the protein synthesis of the host and the virus cells. Okay, we know means various types of cells are present which eventually synthesize various types of protein which are obviously useful for our body. But obviously if that host is being infected by the virus, or we can say the virus infected cell, or simply the virus cell, okay, it will synthesize the protein which is harmful for our body. So it is important to break the mRNA which helps in synthesizing the protein. So the ribonuclease enzyme does that work. So it simply breaks the mRNA of the host and the virus cell, by which it helps in stopping the protein synthesis. Okay, so this is all about interference. These are the antiviral glycoprotein which are secreted by the host cell which is attacked by the virus. Okay, simply the first point, which when it is attacked by the virus. Next, virus infected cell secretes the interferon which diffuse into the adjacent healthy cells, and it activates the antiviral activity of the cell, which eventually prevents the multiplication of the virus. And these are of three types: alpha, beta and gamma. Alpha is produced by WBC, beta is produced by fibroblast, and the gamma is produced by T lymphocyte. And this one is your activity: the interferon diffuse into the neighboring healthy cells, by which it induces the activation of the ribonuclease enzyme 1 in ribonuclease 1 enzyme, which eventually helps in breaking the mRNA of both the host and the virus cells, which eventually helps in stopping the protein synthesis, which is harmful for our body. That is why it stops the protein synthesis of the host cell and the virus infected cells. Okay, so this is all about your today's class, interferon and the types of interferon. So simply today we discussed about in the previous class, as we discussed the external innate immunity, today we discussed the internal innate immunity. Okay, we discussed the elevated body temperature. Now we discuss the interferon. Okay, we discussed the phagocytosis. So this is all about your internal innate immunity, which act as the second line of defense. Okay, also we discussed the acute phase protein. Okay, so this is all about your today's class. We will meet in our next class. Thank you.